Hello guys, welcome to my channel. So as you know, we are studying about the cell cycle class. Right, in the previous video, if you saw the part 1, so we studied about the cell cycle. And these are the phases: M phase, G1 S phase, G2 phase. And we discussed an introduction about the cell cycle control system. So how this control system work? So there are two types of machinery are involved in this cell division. So one that manufactures the new components of growing cell, and the another that hauls components into their correct places and partitions them appropriately when the cell divides into two. So the cell cycle control system switches all this machinery on and off at the correct times, thereby coordinating the various steps of the cycle. So the core of the cell cycle control system is a series of molecular events. So the core of the cell cycle is that it is the series of molecular events that are molecular switches that operate in a defined sequence and orchestrate the main events of the cycle, including DNA replication, the segregation of duplicated chromosomes. Right, these are some of the main events of the cell cycle. All right, so this cell cycle control system governs the cell cycle machinery. So how this cell cycle system works? So cell cycle machinery works like by cyclically activating and then inactivating the key proteins or the protein complexes that regulate DNA replication, mitosis, cytokinases, right? And this regulation, how it regulates, is carried out largely through phosphorylation and dephosphorylation of proteins involved in this essential process. So this entire process of cell cycle control system is based on phosphorylation and dephosphorylation. So what does that mean? I will explain to you. All right, so I will write it down. So first, protein phosphorylation. We'll see that. So what is protein phosphorylation before we move to the cell cycle control system in details? So protein phosphorylation involves the enzyme catalyzed transfer of the terminal phosphate group of ATP to the hydroxyl group of the serine, threonine or tyrosine side chain of the protein. So that is protein phosphorylation basically. So you can see a picture on your screen, right? So that is showing the protein phosphorylation, right? So that what I mentioned. So I will write it down here. So protein phosphorylation, what it has, it involves the enzyme catalyzed transfer, as you can see in this picture. So you can relate it, transfer of the terminal phosphate group of the OTP. So this is terminal phosphate group of ATP to the hydroxyl group. So where the phosphate group is transferred, it is transferred to the hydroxyl group on a serine, either it's threonine or it's tyrosine. And these are on the side chains of this side chain of the protein. So this is basically the protein phosphorylation, right? So this is what you can see, and this reaction is catalyzed by protein kinase. So this reaction is catalyzed by protein kinase. So this reaction is catalyzed by protein kinase, and the reverse reaction. So the reverse reaction means the removal of the phosphatase, right? The removal of the phosphate group, or it's called dephosphorylation. So this is protein phosphorylation, and the removal of phosphate is phosphate is dephosphorylation, and this is done by protein kinase, and this is done by phosphatase. Alright, this is catalyzed by protein phosphatase and this is by protein kinase. So these are protein phosphorylation and this is dephosphorylation, and you can see the picture on your screen. So that shows the how it is, if like the phosphorylation can stimulate protein activity or inhibit it depending on like involved in the site of phosphorylation, right? Right, so why we saw protein phosphorylation and dephosphorylation? Because the cell cycle control machinery depends on the regulation of this protein phosphorylation and dephosphorylation. So phosphorylation followed by dephosphorylation is the most common way, right? That switches, by which this will switch the activity of a protein, that it might be on or off. And the cell cycle control system use this mechanism extensively and repeatedly. So the cell cycle control system depends on activated protein kinases. Activated protein kinases, and it is called as Cdks. Right, so this cell cycle control system depends on cyclically activating protein kinases and they are called Cdks. Alright, so the protein kinases, these protein kinases are present at the core of the cell cycle and especially in the proliferating cell, and it is present throughout the cell cycle. So what happens? They are activated, however only at an appropriate time in the cycle, and after that part is done they are quickly inactivated. So thus the activity of each of these kinases rises and falls in a cyclical fashion. Right, so some of these protein kinases, for example, become active towards the end of the G1, right? So that are responsible for driving the cell into the S phase. And another kinase, for example, that are activated here, that are responsible for driving the cell into G2 phase, right? And so on. Right, so then now the question arises, so which protein or who switches this protein kinases on and off? So switching this kinases on and off at an appropriate time is partly the responsibility of another set of protein in the control system that are known as cyclins. So these are cyclins, right? And what's their responsibility? It's their responsibility is responsible for switching or activating or deactivating, but switching on and off, this protein kinase. That's right. So that's the responsibility of cyclin. So that is why cyclin have no enzymatic activity themselves, but they need to bind to the cell cycle kinases before the kinases can become enzymatically active, right? And that's why this CDK, what does this CDK stand for? The CDK stand for cyclin dependent protein kinases. That is what says cyclin dependent protein kinase. So like for example, this cyclin does not have any enzymatic activity itself. So like for example, this is our cyclin, right? I will represent as a C, and here, for example, in a square, This is our PK, that is our protein kinases. So this cyclin has to attach with this. So like, for example, it may be attached like this. So cyclin is now attached to this protein kinases, and this is how it activates the protein kinase for this cell cycle. Alright, so there are several different types of cyclins, and in most eukaryotes several types of CDKs that is involved in the cell cycle control system. So the different cyclin CDK complexes trigger different steps of the cell cycle. So for example, the cyclin that acts the cyclin that acts in the G2, for example, the cyclin that acts here in the G2, I will write it down just here, cyclin that acts in the G2 to trigger the entry right into the M phase. So G2 is, after G2 it's M phase. So the cyclin that is responsible to trigger the entry of the cell cycle into the M phase, right, it's called M cyclin. So that is called, I will write it down here, M cyclin, right? And the CDK, where it, so the cyclin binds this to the CDK and then it activates and then it moves to the M phase. So the CDK is known as M, I will write it down here, MCDK. That's right, MCDKs, kinases, right? So this is one type of cyclin. There are other cyclins as well called S cyclin and G1S cyclin. So they bind to the distinct another type of cyclin dependent protein kinases, right, for the to move from G1 to S phase or S to G2 phase. Right, so each of this, like for example, this M CDKs or any other S CDKs, so each of this cyclin CDK complexes, so they will phosphorylate a different set of target proteins in the cell. So their main function is to phosphorylate different proteins in the cell, right? So what it does, like for example, this G1 CDKs, for example, phosphorylate regulatory protein that activate transcription of genes required for DNA replication, right? So by activating different sets of target protein, each of the complex triggers a different transition step in the cell cycle, right? So these are the function of this cyclin CDKs, right? Right, so the concentration of a given protein in the cell is determined by the rate at which the protein is synthesized and the rate at which it is degraded. So over the course of this entire cell cycle, the concentration of each type of cyclin rises gradually and then falls abruptly. So the gradual increase in cyclin concentration stems for increased transcription of cyclin genes, whereas the rapid fall in the cyclin concentration is precipitated by a full-scale targeted destruction of the protein. So that means the abrupt degradation of M and S cyclins part away through M phase, so it will separate from the M phase, depends on a large enzyme complex, and that is called an anaphase promoting complex. So down, so that is called as anaphase promoting. So you will have a clear idea when we will discuss about the mitosis, why it's called anaphase promoting complex, right? So this complex, what this complex does, so this complex tags the cyclins, like for example, so this together is an activated Cdks, right? This together, this cyclin and the our protein kinase, this together is an activated cyclin dependent protein kinases, right? So what this complex tags the cyclin with a chain of ubiquitin. If you remember when we studied protein, so how the proteins are destroyed by the chain of ubiquitin, right? So I will write down here. So for example, so when their function is done, so this will be attached with the chain here, for example, and it is known as ubiquitin, right? So this is attached by chain of ubiquitin, and what happens, so eventually after attaching, so this will be like, remember this, just this will be still CDK, but it is inactivated, right? So our this cyclin will be broken in pieces, for example, and this become inactivated CDK, right? So that's the function of ubiquitin. So protein that are marked in this way with the ubiquitin are directed for the proteasomes where they are rapidly degraded. So the ubiquitination and degradation of cyclin returns its CDK to its inactive state. So this is how the CDKs are activated, and then it's inactivated when the purpose is done. So the cyclin destruction, this destruction of cyclins can help drive the transition from one phase of the cell cycle to the next. For example, the M cyclin. So the M cyclin, that degradation and the resulting inactivation of the MCDKs leads to the molecular events that take the cell out of the mitosis. Right, so the activity of the cyclin CDK, this activity of the cyclin CDK depends on phosphorylation and dephosphorylation. So how it depends on phosphorylation and dephosphorylation is because the appearance and the disappearance of the cyclin proteins plays an important part in regulating CDK activity during the cell cycle, right? But there must be more to the story. Why? Because although the cyclin concentration increases gradually, the activity of the associated cyclin CDK complex tends to switch on abruptly at the appropriate time in the cell cycle, right? So what happened? So like what is triggering this this massive increase of the cyclin CDK complexes? So what is actually happening? So the cyclin CDK complex contains an inhibitory phosphatase. What it contains? Where should I write? Maybe here. So it contains inhibitory phosphatase, this enzyme, right? It contains inhibitory phosphatase, and to become active, this complex to become active, this complex need to be dephosphorylated by a specific protein phosphatase, then only it will become active. So thus the protein kinases and the phosphatase regulate the activity of specific cyclin CDKs complexes and help control the progression through the cell cycle. And this is how the cell cycle is controlled by different enzymes and different proteins. So CDK activity can be blocked by CDK inhibitor protein as well. So in addition of to the phosphorylation and dephosphorylation, the activity of the CDKs can also be modulated by the binding of CDK inhibitors protein, right? So this is also how it is controlled by inhibitor protein. This is controlled by the activity can be blocked by CDK inhibitor protein. So the cell cycle control system uses this inhibitors to block the assembly or the activity of certain cyclin CDK complexes. So what happens? Some CDK inhibitor protein, what it does, that for example help maintain the CDKs in an inactive state during, for example, G1 phase of the cycle, thus delaying the progression into the S phase, because maybe the environment is not good, or the environment or internal or external environment is not favorable to move the cell into the S phase. And this is how this CDK inhibitor protein can just inactive the cyclin CDK, so the cell will not move further. Right, so why we discuss these two things? Because so like for example, when the cell cycle control system monitors the cell internal state and the condition of the external environment as well, and before allowing the cell to continue through the cycle, so what it does, it allows entry into S phase only if the environment condition are appropriate, and it triggers mitosis only after the DNA is replicated. So to accomplish all these things, so what the control system does, so it used all these combinations of mechanisms. So for example, at G1 to S phase, as we discussed here, it uses this CDK inhibitor protein, right, to keep the cell from entering to the S phase, and at G2 to M phase, it suppresses the activation of mCDK by inhibiting the phosphatase required to activate the complex, that is mCDK. Right, so this is how. So from G1 to S phase, what it does, it might put the CDK inhibitor protein, right, to keep the cell to move further in here from G2 to M phase to the mitosis if the DNA is not completely replicated. So what it does, it stop the phosphatase enzyme, so this mCDK will not be activated because it has an inhibitory phosphatase, and thus it can delay the exit from mitosis by inhibiting the activation of the anaphase, promoting